Okay, welcome back, class. So we left off on ATP and enzymes. Last part of this chapter is membrane transport. And before we get into the membrane, let's talk about a couple of things. So, making sure I'm on the right side. Remember that the membrane is made up of what's called phospholipids. We talked about this briefly in the last unit. Where phospholipids are these structures right here. Let me get the right pen. Yes, I'm right here. And I'm going to draw a little bigger here. So we have this circle part, and we have these tails. Remember, the circle part likes water. It's called hydro. Oops, that's not an H. Let me go back and get my eraser. Nope, doesn't want to be erased. Give me 1 second. Let's go back to this. We're going back. So remember, while I talk, fix this, I'll explain what I mean. So remember that the membrane is made of what's called phospholipids. Phospholipids, they have what's called a tail and a head. The head, which is the circle part of the phospholipid, is what's called hydrophilic, which means it likes water. And the tails here are called hydrophobic. Remember, hydro is water, phobic is fear, and philic means loving. So these are water fearing. And I say that because that's incredibly important for membrane transport, where the outside is both hydrophilic, where it likes water, but this inside is very hydrophobic. It fears water. It does not want to be near water at all. So because of that, the cells, there's not only certain molecules can go in and out of the cell. So the cell's interior is chemically different from the exterior. The inside of the cell, which is this part, is different from the whole outside here. It maintains this difference by regulating transport of dissolved substances or solutes across the membrane. So solutes are anything dissolved around the cell. Regulating what is inside the cell, an example of homeostasis or balance. So the cell wants to be in homeostasis, so it wants to be in balance. And it does that by regulating what goes in and out of the cell. So the cell membrane forms a barrier. Only certain substances can pass through the cell membrane at a time. And that's because of this inside, like we said, being hydrophobic, not wanting water. Solids can enter and exit the cell by different methods, depending on two factors. And these are going to be important to keep in mind. So one is the concentration gradient, and we're going to go over that in the next couple of slides. And the other is the nature of the um the substance. Is it polar? Is it charged? Its size? It's big or small, and so forth. So we're gonna look at all these today, and we're gonna start with concentration gradient. So a gradient describes the difference. Let me get my other pen now between neighboring regions. And we're thinking about this in terms of the solutes, what's being dissolved in that liquid. Most of the time it's water. And we see in this example, the tea is more highly concentrated near the tea bag than in the rest of the cup. So let me get my blue pen out. We can see in the cup, all this region right here has a lot of that tea, a lot of the tea right there. And this outside part does not. This outside right here, that's clear. So that means there's a gradient with this inside right here having a lot of tea and the outside here not having very little tea. So gradients are directional. And let's write that down or start that. Gradients are directional. This arrow points down the concentration gradient. Since it starts from high concentration and ends at low concentration. So we can see in this image right here that these molecules are kind of moving away from each other. So we slowly have this T spreading out throughout the cup away from each other. We're going from a high concentration, which a high amount of T, to a low concentration, a low amount of T. So gradient, again, is a difference in the amount of a substance. And then there's a gradient always have the directions from high to low. Concentration gradient dissipates without energy input. So concentration gradients dissipate without energy input. They naturally will go away. This is how it looks after time passes. The concentration of T is uniform throughout the whole cup. No gradient is left. So in order to go from the first cup to the next, we see that the next one here, the T is nice uniformly through the cup. There's T everywhere through the cup. So that is when we have no more gradient. The dissipation occurs by random motion. So this gradient going away happens by random motion. The dissipated gradient is an example of increased entropy. And this, I'm not going to have you worry about that term. We're not going to worry about it. The big thing here, know that solutes tend to go down their concentration gradient from a high concentration, which is shown here with this area here having a high concentration, to a low concentration. And that happens until we get no more gradient. That is a very natural process that happens. Another example, and a classic example that I like to use, is say in your classroom, someone in the corner of the room sprays perfume or cologne. Sprays a good amount. In the beginning, if you're on the other corner of the room, you're not going to smell it. But after a while, that perfume or cologne spreads out in the room. It what's called diffuses, it spreads out, and then the whole room smells like that. So that's an example of a concentration gradient, where the first corner where the perfume or cologne was sprayed has a high concentration of that perfume or cologne, and the other corner does not. So the perfume or cologne spreads out from the high concentration in the corner all the way to the low concentration of the opposite corner, just like the C example. So maintaining a gradient requires energy. Maintaining gradients require energy since concentration gradients have a tendency to dissipate. So in this example here that we talked about, the T, we go from the highest concentration or highest gradient to no gradient. So we're going in this direction. And this is a natural process. It does not require energy. This happens on its own. But in order to maintain a gradient, oh, let me go back. In order to maintain that gradient, we require energy. So in order to go maintain the gradient right here, we need to put energy into the system to make sure we can maintain it. So I want you all to keep that in mind. If you go from a high concentration to low concentration, you don't need energy. That's a natural thing to happen. If you go from, if you want to keep a high concentration gradient or build a high concentration gradient, you need energy. Since molecules tend to want to diffuse, tend to want to spread out, in order to prevent them from spreading out, you need energy to stop it. And that's how we get into the different types of what's called membrane transport. So membrane transport is the movement of molecules in or out of the cell, or substance solutes in or out of the cell. We have two types. First one is passive transport. The big thing with passive transport, and this is what I want you to know. Because by the end of this section, you need to be able to compare active and passive transport and know their examples. Excuse me. So with passive transport, the solute is going down its concentration gradient, and this requires no energy. So I will put that in black here. No energy. since we're going down the gradient, you do not require energy. The solids tend to want to go down the gradient from high to low. And then what I want you to know, these are some of the examples we're going to talk about. Simple diffusion, osmosis, and facilitated diffusion. Keyword is diffusion. Diffusion means we're going from high to low concentration. It's a process that's going down the concentration gradient and requires no energy. So if you ever see these words, that means it's a passive transport process. The opposite is active transport. So active transport goes against the concentration gradient. So go back. So passive goes from high to low to low. Active goes low to high. In order to go low to high, this does require energy. So you need energy to go against the gradient. So I like to think of it if you're passive, you're laying down in bed, laying down in sitting down in a chair, you're passive, you're not using energy. And if you're not using energy, you are just going down the gradient, you're diffusing. You're going from high to low. If you're active, you're doing work, you're using energy, and you're pushing against the gradient. You're going from low to high. So if you're passive, you're laying down in bed, you're not doing anything, you're not using energy. But if you're active, you're currently using energy. If you're running, if you're working out, if you're doing some type of physical activity, you're using energy. So that's how you can think about the difference between passive, no energy, active, needs energy. Now, let's talk about the different types of each. So simple diffusion. Simple diffusion is a type of membrane, passive transport. Remember, diffusion means low from high to low. So I'm gonna write that down too. High to low. And the key thing with simple diffusion, it occurs when the concentration gradient dissipates across the membrane. So we can see the example here. We have something go from a high concentration to a low concentration. And the big thing with this is that no protein is required. No membrane protein required. So in simple diffusion, these molecules can go straight through the membrane on their own. They don't need something helping them out. They're going from a high to low concentration too, so they do not require energy. They're using passive transport. And the key thing with this example, if I can get my pen, let's do red, is only small nonpolar molecules can cross the membrane by simple transport. So remember, with simple transport, there's no protein involved, it's a type of passive transport, and it's only small nonpolar molecules. The size and chemical properties of small nonpolar molecules allow them to pass through the membrane's hydrophobic barrier. It is impossible for large polar molecules. So small nonpolar molecules are special. They can go straight through this membrane on their own. They don't need an escort, they don't need help from proteins. They go straight through that membrane. Now, another type of passive transport. This is a special one. It's called osmosis. Osmosis is the simple diffusion of water across a membrane. And remember, it's passive transport. It's going from high to low. So with this, in the example here, we have all these water molecules here. These red circles with these two little dots on them. We have a lot on the left and little on the right. So we have them traveling to the right. Still diffusion, still going passive transport, still going down the concentration gradient without energy needed. But with osmosis, we deal with water specifically. And with water, water moves across the cell membrane, again by osmosis, but it's driven by the concentration gradient of water. And we're going to get three different types of osmosis. The first one is when the cell is in an isotonic solution. In all of these, we're going to look at the cell and the solution, its surroundings, and how it relates to it. And we'll look at the concentration of the solute of what's in the solution compared to what's in the cell. So in isotonic, there's an equal concentration of solutes inside and outside the cell. So that makes the water moving in and out of the cell very equal. Equal movement. The same amount of water that goes in also comes back out. So the cell is healthy, everything's going good in an isotonic solution. Now let's get into the opposites though, the differences. Hypotonic solution. Hypo means low. So in a hypotonic solution, the solution outside the cell, what's happening here is there's a higher solute concentration inside the cell. So we have a lot more sol and molecules in the cell than outside. So what that means is water likes to go to where there's a higher concentration of the solutes. So water is going to rush in and only a little bit comes out. So more water rushes in. You can see this cell here is getting bigger and bigger and bigger, and eventually it can pop like a balloon. So within a hypotonic solution, water goes into the cell. In a hypotonic solution, water goes into the cell, swelling the cell, can make it burst like a balloon. Now let's look at the flip of that. In a hypertonic solution, water rushes out of the cell. Oh, let's get this pin again. So here we have very little solute in the cell. We have a lot on the outside. So as we have a lot of the solute on the outside, water wants to go out of the cell. It's like, hey, I see a lot of these molecules outside the cell. I want to go outside and join them. So remember, isotonic, even movement of water in the cell and out of the cell. Hypotonic, there's low concentration outside the cell, water rushes in. Hypertonic, a lot of solute outside the cell, water rushes out. So osmosis determines water content in plant cells. Plants usually keep solute concentrations inside the cells higher than the outside, so water enters the cell. Hypertonic solutions. So remember, hypertonic means that there's a lot of solutes outside the cell. Hypertonic surroundings result in loss of water, shrinking the large central vacuoles. Remember, vacuole is that organelle within the plant cells that likes to retain water, retain it's help with digestion and so forth. So when there's a hypertonic solution, water is lost, shrinking that central vacuole. This causes cells to lose what's called trigger pressure, which again helps with stability of the cell. We talked about in the last scene how the vacuole likes to be nice and big. It likes to put pressure from within the cell to help the cell keeps its structure. So when that vacuole shrinks, when the cell's in a hypertonic solution, that means we lose that trigger, that pressure, and that causes the plant to wilt. So we can see here in this example, this is the big vacuole here. It's putting a lot of pressure within the cell. But then in a hypotonic solution, it gets nice and small and loses that pressure, which causes the plant to wilt. Now, that was all we talked about simple diffusion, molecules going down the concentration gradient, passive transport, but molecules going straight through the membrane. They're small nonpolar molecules. We talk about osmosis, movement of water. Now let's talk about facilitated diffusion. Still passive transport going from high to low concentration. But the key difference here, which passive transport includes facilitated diffusion, which occurs when membrane protein transports substances across the cell membrane. So this is when we use a protein. When I think of facilitated diffusion, excuse me, it's still diffusion. Something's going down the gradient, but it's being facilitated. Something's helping it through. And what's helping that molecule go down its gradient is a protein. So we can see here, we have these circles going through the protein, this purple structure, down the concentration gradient, and either into or out of the cell. It's very simple, very similar to simple diffusion, but we're using this protein here now. The simple facilitated diffusion requires membrane proteins. The hydrophobic tails of phospholipids in the cell membrane repel hydrophilic substances. Therefore, things like, in this part I'm going to do in red, ions and polar molecules must pass through membrane proteins in order to move across the membrane and down their gradient. So if something is small nonpolar, they go through simple diffusion. If it's ions or polar molecules or bigger molecules, they go through facilitated diffusion because they can't pass through the membrane on their own. So that's a big difference between simple and facilitated diffusion. Both are passive, both don't require energy because the molecules are moving down the concentration gradient. But simple is going straight through the membrane. Facilitated is using a protein to go through the membrane. The last thing we need to go over is active transport. Active transport occurs when the membrane proteins use cellular energy, a lot of times in the form of ATP. Trying to get this my pen to change. Uses cellular energy to transport something across the membrane. This type of membrane transport is often referred to as a pump. Substances move against their concentration gradient. So we're moving against the gradient. If you're active, you're using energy. We're active, we're using energy, so we're using ATP. And we're moving this molecule out. We're moving it against the gradient. It doesn't want to go to the other side of the membrane, but we are pushing it to the other side of the membrane to create a gradient from low to high concentration. So that's why active needs energy. It's pumping something across the membrane from low to high concentration when it doesn't want to go that way. A big example of the pump for active transport is a sodium potassium pump. The sodium potassium pump is a molecule that helps push sodium potassium across the membrane against their gradient. So we can see in this example, we start in the beginning. In step one, ATP binds to the transport protein along with 3 sodiums. So those circles are sodium. ATP binds, ATP transfers phosphate to a protein, which changes the shape of the protein, moving sodium across. So we go from pointing down to pointing up, and the sodium is going out. And when with the protein still like that, 2 potassium ions from outside the cell bind to the protein. So we get these molecules that will bind to the protein. And then I'm going to move this over here. The release of phosphate changes the shape of the protein, moving the potassium back in. So this phosphate group is removed. We go from pointing outwards to pointing back in, and those potassium come in. So the example, sodium, which is the circles, go out. Potassium, which is the squares, go in. and they're both going against the gradient. That's why it's called a pump. That's why it's called active transport, because we're using energy to pump sodium out. We're using against the gradient, and we're using energy to pump potassium in against the gradient. So move this up down here for now. Why does a sodium potassium pump require energy? Refer to the image. Again, like always, take a minute, pause, answer this question, and we'll come back. Okay, let's continue. So with this example, for A, they use the membrane protein. Think about this. Yes, we are using a pump. Sodium potassium pump is a protein, but in facilitated diffusion, we are also using a protein. And in facilitated diffusion, we are not using energy. So that one is out because in facilitated diffusion, we also use a membrane protein. It transports ions, yes, but transporting ions does not mean it has to use energy. An ion can go down its concentration gradient through facilitated diffusion and not use energy. See, it moves the solutes against their concentration gradient. That is the key part, moving against your concentration gradient. In order to move against the gradient from low to high, you need to use energy. So C is the one of why sodium potassium pump requires energy. It's pushing something against their gradient. We got a couple more examples. We got about 3 to 4 more slides. So we got the last couple examples. One is endocytosis. I'm gonna break this word down. Endo means inside. Cyto means cell. So we're moving something in the cell. This is a form of active transport. The cell uses vesicles. Forms vesicles as they engulf fluids and large molecules to bring them into the cell. Substrates can be moved against or down their concentration gradient. So in endocytosis, it's still active transport because we are using energy. But something's going into the cell through is because the membrane, because we're using vesicles. So we can see in this example here, we have these molecules here. We have these molecules here, and this membrane is being pinched. It's going to keep being pinched until we get this circle of membrane that engulfs those molecules, and then that membrane separates. That separation creates what's called a vesicle. A vesicle is just a circular structure of membrane. So in endocytosis, we are pulling these molecules into the cell by creating a vesicle. The cell, think about it this way, the cell is kind of eating. It's grabbing something from outside and pulling it in by making a vesicle. Like it takes energy, so does its active transport. We have exocytosis, which is the exact opposite. And again, I'm going to break down this word. We know cyto means cell. Exo means exit. So something is exiting the cell. So again, here we have cells form vesicles to secrete large polar molecules such as proteins out of the cell. So here, instead of making a vesicle and it pulling something in to eat, to pull it into the cell, we're pushing out. So in this example, this is actually the last thing we're going to go over. We have the vesicle with all these proteins, all these substances. It's going to bind to the membrane here and fuse with it to push all of these molecules out. So instead of pulling something in, the vesicle binds to the membrane and pushes it out. So endocytosis is pulling something into the cell by making a vesicle. Exocytosis is pushing it out by making the vesicle fuse to the membrane and expelling it. So the big thing for this section today And I'm going to put a star next to this. Couple stars. Distinguish between passive and active transport and know the different examples. So make sure you know this. Make sure you can compare and contrast passive and active transport and know the examples we talked about today. Like always, if you have questions, send me a message on campus and I can help you out.